such event I'm participating in. And as you pointed out, I'm going to talk to you today about rickettsias and diagnosing them in wildlife. So if you can see my slides, is the first slide appearing? Yeah, okay, so I'll go ahead then. So the title of my talk is The Birds, the Bees, and the Ricketsias in the Trees. A bit of an intriguing title, and I'll give you the reasons for this as we go through the presentation. So essentially, I'm going to talk to you about tick parasitism of wild birds. Here you can see some Brazilian wild birds with ticks attached to them, and we're interested in the rickettsia bacteria which are inside these wild birds. So tick-borne rickettsial agents in Brazil. Tick-borne rickettsiosis are zoonotic infections which have a global distribution, being caused by intracellular bacteria belonging to the genus Rickettsia. Now, prior to 2004, there was only one species of Rickettsia which had been documented in Brazil. That was Rickettsia rickettsii, the etiological agent of Brazilian spotted fever or febre maculosa. But more recently, during the last decade, an additional 15 rickettsial agents have been diagnosed, infecting a variety of tick species, parasitizing a variety of wildlife, wild birds, mammals, amphibians, reptiles. And from within those rickettsial agents, a surge has appeared Rickettsia parkeri, which is now recognized as an agent of a milder form of spotted fever here in Brazil, in Uruguay, in Argentina, and also in the United States. So it's a field in transition, But in the title I mentioned Rickettsia and bees, So here are some bees. And the reason I'm mentioning rickettsia is because rickettsia aren't only pathogenic. You also have endosymbiotic species of rickettsia. Don't know what's happening here, something strange. We also have jump the slide there, sorry. Here we have some bees. We have endosymbiotic rickettsia, as was shown in this study here of bees, in which these rickettsia play different roles in the biology of the bees. They're involved in behavior, developmental differences between species. Population dynamics can be affected by the presence of these endosymbionts, and even pathogen exclusion. So rickettsia aren't all bad is the message I want to give you with that information there. But do they have something in common? Bees and ticks? Yes. When you encounter them on you, your reaction tends to be this. So they're not completely different. So I'm going to be talking about ticks to you. And in particular, I'm talking about hard ticks, the species of Ixodoideae, which are recognized in Brazil. So there are 48 species, of which 38 belong to the genus Amblyomma, and 9 belong to the Ixodes genus. Those are the two principal genera. However, just this year, an additional 3 species of Ixodes were added to the list. 1 new species and 2 species which previously hadn't been recognized in Brazil. So it's a constant state of flux, this question of who are the ticks which are in Brazil. So rickettsia in the trees, the third part of our title. So here you have the forest canopy in the Atlantic rainforest. And in the Atlantic rainforest, we can expect to find, or in Brazil at least, we find 33 species of Amblyomma. Larvae and nymphs of 23 of those species, or 70% of them, have been found associated with birds in Brazil and in neighboring Latin American countries. So 74% of those tick species were infected with spotted fever group agents. However, there are some important points I have to give here. Rickettsia rickettsii, the agent of Brazilian spotted fever, has never been found in a bird tick. Neither have Rickettsia felis and Candidatus rickettsia assembonensis, which are generally found more in association with fleas than in ticks. Also, Candidatus rickettsia weismannii has never been found in bird ticks. That's found in soft ticks, argosid ticks, isolated from bats, here in Brazil and also in Argentina. And a new species was identified by our group just this year, which is Rickettsia species strain Itingaçu, which is found in argosid ticks, soft ticks, again, this time from frogs. But our focus is mainly on the bird ticks. But what else are you going to find in the trees besides the birds? You'll find other species of animals which are also hosts to these ticks, usually adults of these ticks. And these include porcupines, non-human primates like the black-faced titi monkey, and sloths. So these can also be infected by the same ticks carrying the same rickettsias. So why do bird ticks matter? Research on an international scale is showed here from a group in Illinois that infested songbirds are involved in the spread and dissemination of Lyme disease in the US. Also in Europe, researchers at the University of Uppsala in Sweden, they identified within migratory birds, ticks recovered from migratory birds, they found a newly discovered hemorrhagic fever virus, which originated in sub-Saharan Africa. These birds brought that virus to Sweden. So they're very important for the dissemination of the ticks themselves and possible disease agents. And in the US recently, a group's examining bird ticks which arrived on birds from Central and South America, which within them also had rickettsia species not previously encountered in the US. So when a bird leaves a tree, it takes its rickettsia with it, and that's an important point. So studying this kind of issue of bird ticks and ticks in wildlife, you have to take into consideration there are multiple species and multiple hosts. In addition, Brazil is a country of continental dimensions, which has 6 main biomes, each of which has sub-environments within it. So it's a very complex environment to work in and study tick, animal, and rickettsia interactions. So I'm going to pass very quickly for diagnosis of rickettsial infections in humans and animals, because that's not the focus of today's talk. But if you want more information, this recent paper by Mohammed Abad and his colleagues is fantastic. So I recommend you check it out. It's from there that I take this figure, which shows the main stages when you're trying to diagnose the presence of a rickettsia in a patient, be it a human or an animal, or within an arthropod. Because it's important that we diagnose the presence of rickettsia in the arthropods as well, the ticks. So in the case of human infections, you're more likely going to use clinical data in combination with serological data, and possibly if you have culture or you have material collected from somebody who's in parasitemia, you may be able to sequence and detect the rickettsia directly. But our interest is more on the arthropods and the detection and diagnosis of infections in the ticks themselves. But before I go to that, I'd like to point out that seroepidemiology is something extremely important in the field we work in. It gives us an idea of how prevalent these diseases are and how frequently we're having interactions with humans and these rickettsia-bearing ticks. So there was a study published just this month on human seroepidemiology of rickettsia in Chile, where they found that 5.3% of the rural populations they examined are seropositive for infection by rickettsia. So although the number of cases of rickettsial infection seem to be low, the actual prevalence is probably much higher. It's probably just not being diagnosed. So when we talk about diagnosis of rickettsial infections in ticks, we're really talking about what we call bioecological diagnosis, in which we need to have various components. We need to identify the ticks themselves at the species level, remembering that we're working mainly with larvae and nymphs, which are difficult to identify normally. We also need to identify the rickettsial agents which are circulating, also at the species level. And we also have to take into account the host and environmental factors, which also interplay into our analysis of what this diagnosis really means. So the objective of doing this, and what we try and do in our work, is we try to develop molecular ecological resources which allow us to study the bioecology of amblyomma ticks, our main focus, and the relationship between wild birds, ticks, and members of the genus Rickettsia. Now, the diagnosis of rickettsia in ticks, going back to that figure, I've added a couple of extra boxes here, but essentially with larvae and nymphs, we start with the whole tick, extract the DNA, we amplify for some molecular markers, and then we sequence. That's the normal way we go. We're also interested in identifying the tick, identifying the host rather, sorry, and identifying the tick. And we'll also have information in relation to the environmental data, time of year of collection, altitude, type of environment in which we're doing. Is it primary forest? Is it a dedicated, degraded area, a peri-urbana area? This kind of information is very important in the analysis and the diagnosis we do for these rickettsial infections of ticks. We're not the only people doing this. There's some excellent discussions of this whole topic in the management of arthropod vector data, social and ecological dynamics facing the One Health perspective, a paper published in Acta Tropica in 2018 by Giovanni Benelli and Dr. Duggan. In this system, they have this model here in which pathogen and parasite data, which are important for consideration in model, have to be coupled with accurate data on the arthropod vectors with which we're dealing. Taking them together, we're able to aggregate this data for One Health approaches, which is investigating the interaction between humans, animals, and the environment, the transmission of zoonotic diseases. And although the arthropod vectors seem a little bit out of the three, there's central players in here. And our idea, what we're trying to do with this, is to improve the epidemiological modeling and the prediction of outbreaks. So essentially, we're diagnosing diseases before they emerge. So try understanding the dynamics of tick populations and their ability to act as vectors for emerging pathogens of public health, for instance, Rickettsia parkeri. This forms the basis for the development of risk assessment and intervention strategies which allow you to become tick smart. You can implement educational programs for the local populations to be aware of the risks associated with tick infections. And our project's examining both environmental and animal health topics in an attempt to assess the requirements for seroepidemiological studies of humans and possible animal reservoirs to provide evidence for exposure to the tick-borne rickettsial agents which are circling within the biome which we work with, which is the Atlantic rainforest. So essentially, with our area of research, we're trying to undertake the groundwork, the first stages, the first 2/3 of a One Health problem that requires a holistic approach to be effective. So what's the value of species level identification? Many previous studies on bird ticks and the rickettsias associated with them only identified the ticks to the genus level because it's very difficult using morphological data to identify these ticks to the species level. And molecular examination, as you'll see in a minute, is expensive. So we introduced the idea of you have to identify all your ticks in this study in 2017, which allowed us to identify a novel association between Rickettsia rhipicephala and various species of amblyomma ticks in Minas Gerais. Subsequently, we've applied the same idea to a study of primary forests in two national parks here in Rio de Janeiro state, and we detected an enormous abundance of both tick species and rickettsial agents in association with birds in that environment, which wouldn't have been possible without species level identification. That provided us with the basis to do this study here, which discovered a new species of rickettsia, which had previously been classified as Rickettsia parkeri. It's very similar, but it is a distinct species. And this subsequent paper, which was published by researchers at Fiocruz and from other institutes, and published in Emerging Infectious Diseases, so you get an idea of the importance of this subject, identified human parasitism by amblyomma parkeri, which ticks which were infected with this new species which we had identified. Remembering that amblyomma parkeri is a tick which you find frequently associated both with birds and those tree-dwelling animals that I showed you in the earlier slides. So how do we do this? The classic way of doing it is fairly straightforward and simple. We take a tick, we extract the DNA from it. We then examine it for the presence of one of 4 markers, most commonly the marker encoding small subunit of ribosomal RNA on the mitochondrial genome. We'll separate the amplicons. Should it be the case that we only have a single band, we can then go straight to sequencing, which is the normal step. See here, hopefully your chromatograms will be as nice as that one. And that will give us a sequence which we compare with data sets and we can identify the ticks. Now, but that comes at a price. If you use a third-party sequencing laboratory, as we do, it's 70 reais per sample, which for every 1,000 ticks, which isn't that large a number of ticks in the type of studies we do, you're going to need 70,000 reais to do that study, just to identify your ticks before you do anything else. So obviously something has to change, because Brazil is actually in the middle of a funding crisis for doing research. So we need to find a way to do things more cheaply. In 2015, the government was investing 12 billion reais per year in research. Now that value has fallen down to approximately 1/3, almost a little more than 4 billion reais being invested. But you have to take into consideration that the value of our currency, the real, is probably half of what it was back in 2015. So in reality, we only have effectively a couple of billion reais to play with for everybody who wants to do research here in Brazil. So what we need to do then is possibly go back to the past and play around with what we call retro technologies. Here are some examples of retro technologies for you. Here we have Walkmans, we have Scotch VHS tapes, we have Tetris, and we have the ping pong game, the first Apple computer. So these are described as retro, yeah? And the retro technology I'm going to discuss with you is this here, CAPS, or also known as PCR RFLP. Now CAPS is what? Cleaved amplified polymorphic sequences, or restriction fragment length polymorphism. We combine these two techniques, PCR, with digestion of your PCR product with restriction enzymes, which will cut the DNA in specific sites, breaking it into fragments. So here's an example you see here. You have the PCR product on the left. It's 150 base pairs. We cut it with this enzyme, which cut it in two distinct places, generating this profile of 3 bands. So we delved into the data banks and all the sequences available for 16S ribosomal RNA for these ticks. And we then threw them into this program here, which is NebCutter Volume 2 from New England Biolabs. It's 100% free. This allowed us to cut all those sequences in vitro and generate the possible patterns, allowed us to select 2 restriction enzymes, VSP1 and DRA1, which when used in combination, allow you to produce distinct banding patterns for all the 48 species of hard ticks, which I showed at the beginning of the presentation. There is on occasion a need to use a 3rd enzyme for some species, but generally the 2 enzyme system is perfect. We then validated this system using DNA extracted from adult ticks guaranteed by acarologists as being those species, determining the bands that would be produced there. These matched the predicted banding profiles. And we then tested the system using field material. Initially, we tested it with material which we had sequenced previously, and we confirmed that the banding profiles we'd see would be the same, they were. And then we did the opposite. We identified using the RFLP system, and we subsequently sequenced to confirm the identities. And we had 100% match between the data, so we're very happy with that. And to do the banding profile analysis, we use a program, which is another free program, which is this program here, Gel Analyzer, available at this site here, gelanalyzer.com. Fantastic, so no costs with software either. Now, what are the reductions in cost when we do this, comparing Sanger sequencing with PCR RFLP? Well, essentially, we get this difference here. A single sample analyzed using our 2 enzyme system costs approximately 2 reais in comparison to the 70 reais needed to get the same data from sequencing. So that represents an economy of 97% per sample. Or in other words, for 2,000 reais, we get the same data that we would get by 70,000 reais sequencing. So that's a substantial economy. Now, if we move to rickettsia, the idea is essentially the same. We've identified our ticks. Now we want to identify rickettsias. We homogenize the whole tick again, extract DNA. We do PCR for 4 markers, 2 which are genus specific and 2 which are specific for spotted fever groups. Sequence again, and we'll end up with our identification. Now, with bird ticks, approximately, in many cases, as many as 50% of your ticks may be positive. So if we have to sequence 2 genes from 500 positive ticks, that's another 70,000 reais. So our study is now costing us 140,000 reais. So it's just really too expensive. So even before the fall in financing, our group had developed a restriction, an RFLP, PCR RFLP system for identification of all the rickettsial agents which were known in Brazil at that time, which was 9. So this was the system that we developed here, published in this paper in 2013. We used that continually for the next years, but as we were using that, we began to see that the system would have to be reevaluated and adapt because we have these new rickettsial agents appearing in the story. So once again, what we did was another large-scale in silico digestion analysis. We took over 500 sequences, analyzed them all in the NebCutter program, generated these profiles, and we were able to see that once again, we would be able to separate each of these rickettsia species found in our bird ticks, and even in other rickettsial species. So some validation in vitro here using a 3-enzyme system with the OMP-B target. We're able to amplify all of these organisms here and identify them easily by analysis of our profiles. Some species don't amplify the OMP-B gene, so we have to use the second target, which is the OMP-A target, and that allows us to pick up the rest of these organisms, including Rickettsia parkeri, this agent of the other kind of spotted fever, the milder spotted fever here in Brazil. So what we're dealing with here really is an example of what we call reverse microbial etiology. And there are various examples of this with rickettsias, where Rickettsia parkeri was initially being seen as a ricketsia of ticks in 1937, but was only proven to be a pathogen in the US in 2004, and here in Brazil with strain Atlantic rainforest in 2010. Rickettsia felis, the flea-associated rickettsia, was identified in 1990 and associated with disease in 1994, and Rickettsia massaliae was identified in 1992 and linked to rickettsial disease in humans in 2006. So as Didier Raoult and his colleague Marais said in 1995, the rickettsiosis tend to follow the rickettsiologists. And we're trying to do that now. We're trying to identify and diagnose the rickettsias which are prevalent in given environments, associated with these ticks, understand how they're behaving, and use that as part of the process for diagnosis of tick-borne rickettsiosis here in Brazil. So we come up with this idea here, which is the caveat principle for diagnosis. So your diagnostic system should be what? Cost effective. Ours is. Should be accessible, easily used by almost anybody who wants to use it. It should be validated. Now I've given a plus here slightly lighter in color because we need to do interlaboratory validation with other groups to find out if our system is as robust as it appears to be. Has to be easy to use, has minimal equipment. We can't have expensive equipment because that limits you to laboratories which have the money to buy expensive equipment. Has to be accessible to people who don't have much money to do this research. Has to be adaptable. As we've shown with our system for rickettsia, it is easy enough to adapt your systems as new species appear. And finally, it has to be transferable. The data produced from within here can be switched easily between laboratories. There's no need for you to have specialists for analysis of the data. It's very easy for this to be done. So that's a caveat. But what is a caveat? Caveat in English is a warning. For example, about the use or effectiveness of something. And you have to take that into consideration when you develop a system for diagnosis. So what of the future? We published a paper recently in which we questioned tick-borne rickettsiosis in Brazil. What can be learned from the COVID pandemic? Taking into consideration the fact that COVID emerged from wildlife, it was an unknown pathogen, and it caused a problem. We assume that some of these rickettsias which we see in the ticks will fall into this as well. So here's a warning from the president of CNPq, one of the major funding agencies here in Brazil. And he says that science is not something that you can do without money. And this is our reality. We have to think cost effective. And to think cost effective, you have to innovate. Yeah, here's Blockbuster. I don't know how many of you remember Blockbuster, but Blockbuster didn't innovate and it died. Yeah, so our innovation is what we call frugal innovation. We go back to the past, we take a cheaper technology and we apply it to a problem where apparently only a high-tech, expensive technology will give the answer. And that's not always the case. So we also have to disseminate this information. It's not enough for us to use the traditional form of scientific papers for publication. So to this end, we've developed a web-based tool, which is the Tick Cutter tool, which provides people with access to our database, has tutorials on how to use the PCR FLP systems, has a forum for discussion, allows other researchers to deposit the material there, and we hope that this website is going to serve to re-democratize this area of tick-borne rickettsioses using molecular tools. Because if it depends on sequencing, this area is essentially dead in the current economic crisis which Brazil is passing now, which will pass in the future. So to sum up, what can I say? I can say that the past was very productive. It was productive because we had enough money to do a lot of sequencing analysis, which allowed us to develop these systems. The present is extremely complicated, yeah, more complicated now because of COVID, and the future is highly uncertain, yeah. It's obvious that the only way we can get forward as a country doing high-level science is to work in collaboration. But we have to develop intelligent and workable solutions to do that. Now, hopefully there will be some sun or silver lining behind these dark clouds which we're living now. So essentially, I'd like to wrap up. I think I'm still within time, giving my thanks to the following people. Daniel Paiva for most of those interesting slides and animated slides there. Lucas, who's working on the improvement of our website. Dr. Tassia, who helped me a lot with the sequencing analysis. Professor João Facini and Professor Hermes Luz, who opened the doors for me to work on this area of research, which has been fantastic during the last 7 or 8 years. I'd also like to mention a group which was formed because of COVID, the debate group called Carrapatos Pandemicos, where we discuss all kinds of topics around this subject of tick-borne rickettsioses. And it wouldn't be fair to finish this presentation without thanking Doctors Aber, Nathans, and Smith, who developed restriction enzymes, and Dr. Kerry Mullis, who developed PCR, as well as the different funding agencies and my own institution and postgraduate course, which have given me a lot of support during all of this 6 or 7 years of research I've been involved in. So thank you very much. I'll end there.
Thank you, Dr. McIntosh. Thank you for your excellent talk. We have here one comment from Teca Galvão. She's saying, the identification of non-tuberculous mycobacteria by reference laboratories is often done by the same type of technique, PCR, followed by digest. So this is actually quite relevant in real-life scenarios for other types of pathogens. The only issue is having enough diversity in the sequences, which you have shown is here.
Yes, we didn't expect to find a single gene within 48 species of ticks that's only 460 base pairs in size, which would allow us to identify all the ticks. That was a very, very welcome surprise. There are some situations where you may have a doubt, but we can simply, entering with a third enzyme, where we have this, it's a deal between 2 species, using a third enzyme system, we can resolve it. But yes, this idea of restriction fragment polymorphism, PCR-FLP, it's not new, it's retro, it's forgotten. Most students who enter now to do molecular biology courses, they want to know about next generation sequencing. Many people, when you talk to them about PCR-FLP, they'll tell you no, because it's very difficult. But it isn't difficult. It's fantastic. It's like doing a jigsaw puzzle, putting those fragments together. And with the free programs which we have to do this, anybody can do it. So instead of paying, let's say, 200,000 reais for one project using NGS for this analysis, why don't we fund 4 or 5 projects in different places around the country using the PCR-FLP method and generate lots more information from different biomes, different tick species, that's really going to be useful for us to determine what the real importance of these tick-borne rickettsias are, which there are many, many. Many of them are probably endosymbiotic, but some of them have the potential to be pathogenic, and we need to be aware of this. It's an underreported disease. Any more questions?
Yes, there's some more questions. Helena Sage is saying that this methodology is used for primary identification of NTM. It is a nice methodology. What is the frequency of samples with mutations losing the restriction sites in with the same parts?
Okay, so from our 48 ticks, we had 48 tick species. We had 2 tick species which were known to have high levels of variation in their 16S sequences, which are Amblyoma ovale and Amblyoma parvum. Now, each of those generates 3 or 4 possible profiles in our tick identification system, but those profiles seem to be stable. We're not identifying a lot of interspecies variation. Only time will tell, Elena. We have to check more. The same with the rickettsias. We haven't found much evidence. We've examined more than 3,000 or 4,000 rickettsias now by this system, and we haven't found much evidence of misidentification with the system. So, and none that we could ascribe to particular mutations in the restriction sites. But that's obviously a risk. The system has to be adaptable. That's why we have this website. We want the website so that if somebody finds a variant, that sequence variant goes into our data bank and we find a solution for it. There has to be a third enzyme, because if it's mutated, it'll probably give a distinct profile that doesn't match with any of the other profiles that we're looking at. Does that answer your question? I hope so.
I think you did well, Dr. McIntosh. We have some comments from one congratulation from Dr. Caetano Antunes, who is a professor from
Okay, thanks,
Fiocruz as well. He's saying that it's really cool work and great example of how ingenuity can help us more forward.
Yeah, that's true.
And then Ana Paula is saying that, do you think it's possible that this methodology is transformed into a product to be used in public health laboratories?
We have spoken to Gilberto Gazetta, who's from the reference laboratory at Fiocruz. He was a member of the examining board of Daniel's thesis, Daniel Paiva, who developed most of this work in my laboratory. And they're very keen to have a look at this, because they know they have this problem. They collect thousands of ticks, and not having the ability to sequence all those ticks for financial reasons, which is increasingly the case, is, it's a huge barrier to anybody who wants to get involved in this kind of research. So you have to have alternatives. This is a viable alternative. May take a little bit of a mindset change within public health laboratories, but it's a kind of back to the future approach. This is actually had a project now which was just funded by FAPERJ. The title of the project was exactly this: Back to the Future, using low technology molecular approaches to provide high quality genotyping and molecular data in parasitology. Because PCR-FLP isn't the only method. You can work with DGGE, you can work with high resolution melt analysis. If you have a machine to do real-time PCR, but you don't have the money to buy the reagents to do real-time PCR, instead of that machine sitting there doing nothing, you can transform it into analysis of your PCR products using high-resolution melt analysis. Very cheap method. It's coming back in vogue as well. Retro great, I'm all for it. I'm all for Tetris, I'm all for VHS. Bring all these things back. Go back to my youth.
Yes, Professor. I think we have one last question that's from myself, and it's about the ticks.
Okay.
Is it totally necessary to use the molecular methods for tick identification? What about the morphology?
Okay, well, in the case of bird ticks, most of the ticks we're going to be dealing with are larvae and nymphs, primarily larvae. And there is no identification key for larvae of amblyomma. There's one being developed by Dr. Thiago Martins at USP, and that will probably be a big help. There is a key for nymphs, but you really have to be a highly trained acarologist to identify these ticks by morphological methods. There's a requirement really for molecular diagnostics, and sequencing, it's out with the reach of most laboratories which are in these biomes, Amazon, Mato Grosso, the Caatinga, these biomes here. These laboratories don't have the money to sequence all the ticks. So morphological identification is fundamental. We always identify all our ticks to the genus level by morphology. But we have Dr. Fassini and we had Dr. Luce, who are two excellent acarologists. If you don't have an excellent acarologist, you have to go for molecular. It's as simple as that. Adults, less difficult, but still very difficult. So hope that answers the question.
Well, if there are more questions, people can leave the question and then you